So Sylvain Costes is Space Bioscience Research Branch Chief Acting at NASA Ames Research Center. He is the Principal Investigator in the Space Bioscience Research Branch at NASA ARC and the Genelab Project Manager. Dr. Costes has spent his career leading multidisciplinary research in both academia and government scientific institutions. He is coordinating the systems biology scientific research effort for the Genelab project, supporting both internal and external collaboration efforts, and is representing the project in support of the Space Biology Project and the ISS program. Dr. Costes attained his PhD in the Nuclear Engineering Department at the University of California, Berkeley, where he studied radiation biology. So today he's going to talk about the open science effort to foster space exploration.
Well, thank you, Mark. Can you hear me? Can you see my screen?
Yes, yes, we've got both. Thank you.
Perfect. Okay, that comes up. Let me take this out of the way. Hold on. I've got my face in front of me, so I can't see my screen. I don't know if you see that little window here. Maybe I can.
So it's on the first slide. It says space biology, understanding fundamental biological responses to the space environment.
Yeah, I have just my face. Hopefully I'm not gonna kill it. Okay, perfect. I was hiding my webcam. Sorry, guys. Well, thank you very much for the intro. And I think this talk is going to be a bit less technical and a bigger picture of what we're trying to achieve in space biology program. So just to dive into some of the concerns we have when we go into space are listed here. We call them the space stressors. You probably have all heard about the radiation exposure and the altered gravity. They are the two main stressors that are pretty well characterized. But there are other things like harsh temperature and atmosphere, pressure. In terms of the sleep cycle is quite altered through these light cycles. You rotate quite a bit around the planet during a 24-hour cycle. That can really affect the sleep pattern of our experiments, including animals and humans. And for humans, I think the isolation and the closed environment is a concern. For other things, such as going to the moon and Mars, the regolith is the dust that's found on these planets or satellite, and they tend to be quite well. We are still studying what they really do to the organism. And so one way to deal with this is to really let's see if it's yeah, perfect. So the way we deal with these questions is to run as many experiments as we can on different platforms. Right now, the main platform is the ISS, the International Space Station. It's a laboratory in space. You probably have read some of the news, like we use the MinION to do sequencing in space. That was done a few years ago. We can do PCR, we can do sample prep, we can do imaging microscopy. You can take images of bones in space. We grow plants. Quite a few experiments are taking place in the biology field. And then, one thing we're really trying to add to this aspect of the work is really the concept of open science. It's fairly new for biology in terms of space biology. It's a well-known idea for other activities at NASA, such as, the Hubble telescope, and open science has been really pushed very far with these groups, with these physicists. But in the biology realm, there's a tendency to work into a bit of a, in a little environment. You tend to encapsulate yourself into your own research, and that's a culture that we need to change. So I am part of the open science effort to change this culture. So I use here the definition that we use at SMD, which is our directorate, the Science Mission Directorate. So our division is part of this big group of divisions, including astrophysics, heliophysics, and so we're lucky to work with these big divisions. And we define open science as a collaborative culture enabled by technology that empowers the open sharing of data, information, and knowledge within the scientific community and the wider public to accelerate scientific research and understanding. And I think, AI ML techniques are going to be playing a huge role in this open science work for space bio. Now, This is kind of like and it's a very simple cartoon, so I apologize for the simplicity, but it gives you an idea of where we are right now. So currently experiments come from two kind of platform. We have the analogs, which are anything that simulate what we see in space, and then we have actual space data, and mainly ISS, but satellites can also be involved here. Experiments are collected, and then we have a database that stores the data. If it's omics data, so proteomics, transcriptomics, it goes into Genelab, which is one of my projects. I'm the project manager for Genelab, and then I'm also the project manager for ALSDA, which is the non-omics database. So anything that is non-omics, like images, phenotype, anything that is not omics goes to this other database. PSI is a database for physical experiment, so I won't talk about this today. But they are part of this open science repository data that we have. The PI plays important roles in entering the data, but there's also other way of entering data. We have this archive system where tissues are being collected and shared. So if an experiment is done on a mouse and the mouse is dissected, every tissue is, not every tissue, we try to take as many tissue as possible, leftovers from the PI, from the principal investigator, and they are stored into this archive at NASA Ames Research Center in California, and someone can put a proposal and ask to do more research on those tissues. Our team itself, Genelab, is doing some of this work. We have some budget that allow us to also study these tissues. And then the user community is very important here. So we have our main users working with the PI, and then we have this citizen science group, and we also have a strong bootcamp training system for high school and universities. So we do quite a bit. This is one of the analog for space radiation, is actually at the NASA Space Radiation Laboratory, which is at Brookhaven National Lab. So if you think about what we provide in terms of open science, we have become more of an open science enterprise solution. And Genelab.nasa.gov, I encourage you to go check us out. This is really the main repo. ALSDA built on top of Genelab, but Genelab was the core repo. So we started with the omics, and now we're generalizing our approach to non-omics data. And then so we have a repo system. We have a workspace where PI can share and store their data before they want to make them public. It becomes kind of like this Dropbox idea where you can work and organize your data before you submit them. We also have a different visualization tool to look at the environmental data of an experiment. So those are experimental metadata like temperature, humidity, CO2, high radiation level for a given experiment. And we have a visualization platform and an analytical platform. On the back end, we don't use SQL, we use MongoDB, we're on the cloud. We have quite a few API, and we federate search and across a bunch of different databases such as EBI, NIH, and so on. In terms of sources of data, we have quite we generate our own data, which make us quite unique compared to NIH or EBI. We make data as well. So we have a sequencing group that basically take this sample from space that are leftovers, and we run them and we put them online for anyone to analyze. So they're free data, untouched. But I think that summarized very well what we've done. We are very proud to say that we're one of the most fair database in the world right now. We had an advantage over NIH or EBIs that we started way after. So we built Genelab with fair in mind. So it was really built around this concept. So that made things easier. We don't have to go back and fix certain things. So, let's see. Is it going to go to the next one? Okay. So one thing we've done, I'm sorry, for some reason, this software is kind of slowing down my computer. So I'm just going to go through quickly. One concept that we pushed, we created those analysis working group. It's a scientific community. They join us, on a voluntary basis. We meet monthly. We have 5 different groups, 4 groups focused on animal work, multi-omics, microbial analysis, plant work, and then we have one on non-omics, so ALSDA group. So overall, that's about 300 people. And then together, we've been coming up with ways to turn these raw data that we have in our database into something that makes more sense. So for instance, in the case of gene expression, you go from raw sequencing count to, a disease state. We heard some of that today through pathway analysis and so on. So we came up with a consensus pipeline that basically we all agree on out of the 100+ people that this was the best pipeline that we thought. And then we vet that pipeline and we process all the data systematically through it. And the goal here is to really not give access to only the data scientists who can do this work on their big computer and on having the right knowledge, but it's really opening the door to citizen science. So again, there's two big groups that we call them the super groups. The analysis working groups is one of them I just mentioned. And then the education working group is really the one using some of this pipeline, some of this information to really teach concepts. One thing that was pretty clear when we did this work is that in 2019, this is when we opened the access to process data through our visualization tool. And the moment we did that, over a month change, that was when we released it in October. By November, we doubled the number of access to our repo, and that was really the majority of the access was the process data. So, going from raw to process really doubled almost overnight the number of downloads. So those are my two big power user group, the AWG and the EWG. For we have 48 universities in the US joining us, 4 NASA centers, several countries. And so anyone here in this talk, in this group today who's interested to join us or collaborate with us, please reach out to me or talk to Mark, and he can send that information to us, and we'll be happy to have you join one of these groups. We teach, as I said, and as a result, we had a big package in sale that came out last year in end of 2019. It was a package of 29 papers, I think. Yeah, 9 of them were from data that were generated by Genelab or using Genelab, and then this was really the result of this AWG. So that was a huge ordeal for us. 29 papers in one shot in the cell press with quite a few in the actual cell itself, and then cell report I sent. So I would highly recommend, if you're interested in the biology of spaceflight, to go check it out. I'm listing also. So we have a lot of let me go over this graph here. We call it return on investment. So basically in blue, you see the original publication in our database that are going up every year. So every time you submit the data to our system, typically there is a publication associated to it. What you see in red is the number of publications that are derived from one or multiple data sets. And so we've been tracking them separately because they were not the original publication from the PI. Those are typically, a lot of them actually are driven by the AWG, but what you can see is that our trend of derived paper is going much faster than the number of papers. So, and that's very encouraging. On the right here, you can see the different, type of data we have. We are mainly transcriptomic, and that's because this is probably the most mature field out of all the omics, with RNA-seq taking over macroarray data that are older data. But we are covering the full spectrum of omics. So, and then we're also covering the full spectrum of organisms from humans. No astronaut data in Genelab, unfortunately, except for a few exceptions of data that were already available online, but we are not authorized to put American astronauts on this database. So it's going to be, if it's human, typically it's human cells. Let's see if he wants to go to the next one. Sorry. So in a nutshell, we have an open science submission portal that both go to omics and non-omics. And then now I want to emphasize a bit about the topic of this group, which is, how do we model things? And we've been looking at, in the past, we've been looking at modeling things through classic pathway analysis, but this has really changed quite some in the recent years. And we now have an Aiml group, and actually have the two main participants of these groups are in this talk today. So I'm sure they can engage in questions and answer if you have. But we focus on two main techniques right now: knowledge graph models and causal inference models. And there's one paper on the knowledge graph model. I'm going to go over these two techniques very quickly. But the idea is to create this. We're very much keen on creating a portal that would really be living off people's work, like what we heard in this talk today. We would love to see the integration of some of your tools into our system, just like we've done with others. And I will show you what we've done so far. So we're not necessarily building these tools. To some extent, we do sometimes, but we mainly use them. Our goal is to have a knowledge-based predictors, and we hope by October of this year to have our new Genelab Viz 3.0 that will have some of these AI/ML capabilities. This is our current Viz tool. We can basically visualize data set, PCA plot, volcano plot, differential gene expression, the classic things. This is an interesting one I like, the PCA plot. I use that more for people who are not so savvy in analysis, but you can see here actually a radiation response to an experiment, and how on the PCA plot this distribution clearly shifts. So you can see how radiation, increasing the dose of the radiation to a tissue, moves the position of your vector. So interesting visual here. We have the same concept right now for non-omics. So here this is looking at multiple data sets at once. This is our goal. We're not really interested in analyzing one experiment. We want to look at multiple experiments. And again, when you do that with classic pathway analysis, one of the challenges with this is that the normalization scheme is extremely complex and is never quite good. And AI/ML tend to have is a much more lenient in terms of taking heterogeneous data set and not having to deal as much about this normalization scheme. So here, this is a classic distribution of changes in bone densities. So you can see here values of this is flight versus ground control. So animals in space versus animals on the ground for a bunch of different experiments. Those are old experiments. STS is space shuttle, Bion is a satellite, SpaceX is more recent. And overall, what you can see is those mice in space have a lower densities. And so that, this is the kind of the things we want someone to go on our website and within a few clicks be able to see this, and so we're working hard on this. This is not created yet. It's really a concept right now. We're still thinking about this. Okay, let me dive into AI/ML. I'm moving very fast because I think we're a bit behind schedule. So Mark, interrupt me whenever I'm.
We've got plenty of time, don't worry.
One thing we've done with this AI/ML group is I'm going to cover really two, just for time's sake. I will just talk about two techniques that we developed, or one that was developed by NASA and one that was really integrated from UCSF. So for CRISP, I think I mentioned the CRISP technique. Let me go over. The idea was to use multiple data sets. In this case, we had 3 human data sets and 3 mouse data sets. All these animals had been flown in space. I'm sorry, they have been exposed to radiation. So it was analog experiment where you just see what radiation does to a living organism. One of the first tasks was to basically put everything as a human. So we did a human mouse gene homolog mapping so that we can all call the same genes the same thing. And then we use this technique called CRISP, which I don't know how much you heard from my first slide on FDL, but basically it's a technique that was developed by this annual contest. It's a 10-week process where people apply. And so it's a second iteration of this tool. The beauty of CRISP, it's a causal inference model. We can take any type of data. You reduce them through dimensionality reductions to remove anything that's being duplicated, basically. Then they use different causal inference modules, and we have then this ensemble model that is you do a voting ranking based on what is the model that is, what is the path or the signal that is the most preserved across all the different models. So you have these voting techniques. So in our case, like I said, we had 6 data sets, 3 mouse, 3 humans, and the targets was either you are or you are not irradiated. That was the given target. And then what you can do is you can use CRISP to identify the features from the entry that are causal of the target variable. And doing this was really remarkable is that we picked many cancer genes, which is something we know, radiation induces cancer. So, but it was not something we could see that well when we looked at individual experiment. That was the interesting thing, is when you put them together, we see a better response. There's a lot more to it. And the team is actually preparing papers on that. So I don't think it has been accepted yet. So that was one aspect of the work. On graph knowledge theory, we actually collaborated with UCSF, and we used the tool called Spook, which seems to be very similar to Crossbar. And then here, the two nodes that I think are really interesting is gene and anatomy. So basically, each of these nodes is a database. And let me show you the power of this in our context compared to classical techniques. So here again, we went into Genelab and we used 6 different data sets, thymus, spleens, and liver. And those animals were flown in space on the ISS for different duration. And there was a ground control. So you go from 13 days to all the way to 60 days animal experiment. And if you do a PCA plot, we see that the tissue, there's really a very strong cluster by the experiment and by the tissue. And then if you look within each of these clusters and you look at the 3 groups, which is a ground control, a space flight, and the basal control is the animals before they were flown. So that's kind of your time 0. It's really hard to see a strong separation within those clusters. Worst is that if you want to look and do a meta-analysis and you start looking at the pathway overlap between the thymus, the liver, and the spleen, well, there's almost no overlap. So that's kind of frustrating. Now, if you use instead Spook, so Spook used this concept of a vector. They basically vectorize all this gene expression and also the anatomical information, and they then run it through the graph knowledge system. And if you look at one of the nodes, so I took that node because I find it fascinating. It's the symptom node. What you see here in different colors is blue is ground versus baseline. And then this one is the one that are really important is space versus baseline and space versus ground, because we expect to see some changes from space. And so those are the green and orange. And you can see indeed that in this response, we have a nice separation. And what was interesting is we picked up, for instance, space motion sickness and some disruption of the hearing, which is 2 phenotypes that astronauts have been mentioning in space. And again, here we're only looking at mice, not at human. There's no human data in there. 1. 2, we don't use any brain anatomical data or eye or hearing. The tissue were thymus, liver. What was the 3rd one? Anyway, and then what you see here is that the nodes that are picked up actually pick up brain and head and so on. So pituitary. So even though this is really the power of these techniques compared to the classic pathway analysis, and to me that was kind of like a big moment. I was like, okay, this is really taking us to the next level in terms of analysis. So I'm super excited about it. So we're really working hard now on integrating these techniques. To summarize our challenges, we have low number of samples. We typically can only fly 20 mice in space at a time. We have high dimensionality data, as most of you guys, 50,000 genes versus 30 samples. You can see the challenge right there. We have heterogeneous data. We have many different factors. And so we have to deal with all these things. So this is actually right now what we're doing for AI for LS activities. We're doing a benchmark data set. So those are going to be data set that can be used for machine learning training from the rest of the world. So people will be able to go and look at classic samples that have some kind of importance for us. So one will be images of human cells exposed to radiation, and we're looking at the DNA damage. One will be RNA sequencing data, and part of that will be fake data using GAN. And then what is the next one? And then we also have CT images of bone of animals that have been flown in space, and you can see the deterioration of the bones in space. And so there's a whole catalog that's being created. So it's one of the main activity we have right now. And we're also working on integrating. I'm going to go, because I have 10 minutes. I'm going to go quickly to, this slide. This is the, we're working very hard on basically integrating these different tools, CRISP and Spook, but Crossbar or anything else you guys are interested in trying to integrate into our system, please reach out. I have both Lauren and Adrienne are in this call, and Adrienne is actually in charge of the modeling hub for Genelab. So you can put you in touch with her if you're interested in trying to integrate some of your tools and see how we can bring it in. This is supposed to come out on October 1st of this year. That's our release time. I want to finish by saying that we had a big workshop in June 2021. Actually, some folks from Biorelate joined the workshop. As a result, there's 2 papers being submitted to NMI, Nature Machine Intelligence, and we also have a white paper for the Decadal Survey, which is every 10 years, the National Academy of Sciences is contracted to go over a slur of white papers submitted to the Academy about what we should be doing for space bio at NASA. And they review all this information and they give their guidance. So this is going to happen in 2023. They're going to review all these white papers. We put in one paper on AI ML for space bio. This is available online. You can read it if you're interested. And for the NMI paper, there's 2 of them. And you can also we can read them once they're being accepted. So we're waiting for what they're going to say. So on this is the team. Unfortunately, I don't have a recent picture because of COVID. So the last picture is 2019. So we need an update. This team has changed quite some. And a big thank you for the AI for life science team and specifically Adrienne and Lauren, kind of like basically leading this effort. Thank you.